So in order to bring us into hemispheric lateralization, I just want to sort of address this cultural understanding we have of the right and left hemisphere. If you've ever heard the phrase, I'm very left-brained, I'm very right-brained, I'm very artistic, I'm very analytical, that has a little bit of basis in evidence, but not really a lot. There's nothing that says a right-handed person can't be artistic or that left-handed people will be artistic. That's quite an exaggeration. It's a very aesthetically pleasing exaggeration, as you can see here, but it is not clinically accurate. So hemispheric lateralization, you do have some very specific things that are lateralized, and we've actually already talked about some of those things that are lateralized. For example, where are Wernicke's and Broca's located? Which hemisphere? They are only in the left hemisphere. So you will have regions in the right hemisphere that are similar and analogous, and we will talk about what those are. So they're going to be complementary but different functions. Then I just want to point out this vocab word up here, patellus. It's probably pronounced a couple of different ways. Humans are not symmetrical. Most of us are shifted. Our skull is entirely shifted with the right side usually a little bit more anterior to the left side. That's our entire skull, and that also feeds through to our brains also being a little bit shifted. So that's what patellus is. It's just that shift. If you've ever flipped over a picture in your selfie camera and been freaked out by how asymmetrical you are, it's just because you're used to looking at your skull in a certain way. We are all asymmetrical. That is the normal state of growth where things shift. No, so again, both hemispheres have those higher order centers, but we do have lateralization. We do have some unique things. So Wernicke's and Broca's are unique to the left hemisphere. Obviously, there are some disorders that would reverse this or change this, but for most people, Wernicke's and Broca's are going to be on the left. So we do call it the categorical hemisphere, and it's just an easy way to sort of narrow things down and simplify it a little bit. So the left hemisphere is categorical. It does tend to be a little bit more analytical in reasoning tasks, but I do consider this a little bit exaggerated for what the brain's actually doing. In contrast, the right hemisphere is generally considered the representational hemisphere. So it's going to be a little bit more involved in visual-spatial relationships and analysis. And we do say things like music, interpreting music, interpreting artistic skill or having artistic skill. Again, this is a little bit exaggerated in my opinion. So while you have Broca's and Wernicke's on the left, your analogous regions on the right side would be things like if you're understanding speech over here, maybe you're understanding sort of the emotional content of speech over on the right side. Maybe you're understanding lyrics to songs over on the right side. Things like that. So it's complementary function. And then in order to access both of these, we need commissural fibers. In order to integrate these two ideas, we heard what was said, we understood the words, and then we understood the word meaning. That means we have to have a connection between the right and left hemisphere. That's commissural fibers, that's the corpus callosum. So in general, as an average, women have a proportionally larger corpus callosum than men. So by proportions, not overall, right? So we do say usually women have better lateralization than men. That's one of those changes. Men overall have a larger brain, which goes with a larger body, right? But women will have a more foldy brain with a larger corpus callosum. So the way that I explain this to people, if we've got understanding of speech and then emotional content of speech over here, is understanding the difference between do whatever you want, honey, and do whatever you want, honey, right? Good times, good times. Yeah, so that's about it for hemispheric lateralization. Yeah, question. Mm-hmm.
But because the musical abilities on the other side,
you can still sing
words.
Yes.
And actually get their and then by using that and adapting that, they can teach themselves to talk again by using that side of the
right. Just be more musical when you speak. Yeah, absolutely. Yeah, and that's part of how we figured this out.
Right?
Mm-hmm. Yeah. That having been said, learning song language or song lyrics in another language is a terrible way to learn a language. If anybody's ever tried to learn a language, oh, I'll just listen to the music and I'll translate the song lyrics. That is the least efficient way to learn a language. Don't do that. Okay. So again, lateralization and in being able to access both hemispheres relies on commissures, especially the corpus callosum. It's the only one that matters to us. And again, lateralization correlates with handedness. So people who have who are left-handed will have a slightly different lateralization pattern than right-handed people. I've never seen the clinical outcomes for that. I've never seen the research into that, so I don't really have a comment into it. I don't know how true that is. And then it doesn't say anything about ambidextrous people, so. All right, let's talk about memory. I took an entire course in memory, so we could go all day on this. I have added some stuff to this compared to what your textbook wanted you to know about memory to make it a little bit more useful for you. It is based on storing and retrieving information. That means we're going to have some complex circuitry to get to memories. We've already covered parts of the limbic system, and especially the main part of the limbic system that we actually need. What part of the limbic system has to do with memory? Not the hypothalamus. It is an H word. That's why you're doing the hippocampus. Now, does the hippocampus store memories? What does it do? It files and retrieves them appropriately, right? So it's actually going to activate where the memory is actually stored. Nope, that's emotions. On the hypothalamus. It's in the. I don't know what you're saying. Sorry. In different parts of the neocortex, in your association cortices. Remember, you've got your association motor cortex that understands what you do and stores memories associated with motor, memories associated with sensory, memories associated with visual, memories associated with auditory, so on and so forth. So we're actually going to activate all of these different neurons and all of these different cortices to activate that memory. But the hippocampus is the one who is activating the correct ones. So here's your textbook content on memory, your different types of memory, and this is all episodic memory. This is your memory of episodes as opposed to motor memory, your ability to physically do things. So you'll have sensory memory. You pick something up and you have that sensation and you'll remember that sensation for a matter of milliseconds. If you happen to pay attention to it, you can move it into short-term memory. And if you happen to repeat that information, then you can encode that memory into long-term memory. And the bad news here is that you can forget literally at any time. Yay! So in order to develop a memory, you must encode your memory, and encoding requires proper functioning of the hippocampus. And the amygdala, what's the amygdala doing again? The emotions, right? You've got to have an emotional component to that memory as well. All right, that's what your textbook gave you. I'm going to give you some more useful information about what's actually happening with memory formation. So you'll have a connection between the hippocampus, the amygdala, the hypothalamus to link memories to specific emotions. You'll have the hippocampus forming and encoding your long-term memory. When you're encoding, what you're doing is specifically developing new synapses. So we have a saying that neurons that fire together wire together. So you'll have a different circuit for different memory. What you're doing is you're activating different cell bodies in different parts of neocortex in the association areas. And we actually do find these are pretty consistent between individuals. If we show people this picture of Mark Hamill, we'll get certain neurons activating within the cerebral cortex. I've heard this, there was a whole paper written on the Jennifer Aniston circuit. We all have a Jennifer Aniston circuit. If you know who Jennifer Aniston is, the same basic circuitry is firing in your brain. And that means you've developed specific synapses. So there was no synapse, and then you developed a new synapse. And you strengthen that synapse through repetition. Yeah. Yeah. In fact, I might be able to find what the Jennifer Aniston circuit looks like. This was a famous paper. Let's look for it. Aniston, is it 1 N or 2? Aniston circuit in the brain. Yep, came right up. Is there a picture? No, of course not. It's a images. May not be able to find. I like that picture right there. That's good. It's exactly what it looks like. No, I'll track it down during our next break if I can. There's a specific circuit. Okay, so to learn, you need repetition. At the end of the day, all you need is repetition. It is actually possible to interrupt the formation of new neurons or old neurons. We actually have the ability from science to destroy any established memory. We know because actually every time you activate a memory, this is where it gets super weird, you actually destroy that synapse and rebuild it completely from scratch every time. Yeah, that's a big part of it. And again, this is a little beyond what you guys need. It's just really clinically interesting. Your hippocampus is the only part of your brain that has mitotic neurons. Remember I told you adult neurons are non-mitotic with very few exceptions. The exceptions are in the hippocampus. You are growing new neurons in your hippocampus. So yes, your memories do change. Your memories are very volatile. They are not stable. Human memory is terrible. It's really bad. And you can mess with it. There are ways to just stop it from working entirely. So that's what I wanted to convey to you. You need repetition. You need repetition. You need repetition. No, absolutely not. And by the way, encoding happens during sleep. REM sleep is going to allow you to encode things into long term. Without REM sleep, you really won't encode things into long term. Yep.
Cramming can help for it. You can cram and take it right away.
Yeah, your short-term memory is very limited. Your brain does like sets of 3. If you want to utilize this, the max you're getting is 3 sets of 3. That's the max you can get out of your short-term memory. I would not recommend it. Yeah, I mean, you can cram, you can maybe pass that test, but you're not going to encode it into long-term memory. It's not going to work. It's way more functional to study an hour before bed every night than it is to cram for 12 hours and then take a test.
The best time to study is right before.
Probably, yeah. We've done specific studies in rats. So there's a rat study that I talk about in my sleep lecture that is a couple of different groups of rats. They all have to run a maze. And then one group is allowed to sleep. Another group is deprived of non-REM sleep. And another group is deprived just of REM sleep. And the rats deprived of just REM sleep, they see the same maze the next day. They are starting from scratch. Yeah. Whereas the rats who got either full sleep or were deprived of non-REM but allowed REM sleep, they have learned the maze. They go through it much more effectively. So get sleep. Sleep counts as studying. Just right now. just take a nap right now. That's the way to do it. Yep. And another thing that I've added into here is that is episodic memory. Procedural memory is different, which again, I think this has a lot of relevance to you as nurses in training. You're going to learn a lot of physical tasks as part of your nursing training, and maybe you'll get tested on it in an episodic way of, okay, now what do you do next? What do you do next? That is not nearly as useful as the clinical experience, the lab, the sim lab experience that you're going to physically get to be able to do that task. And you can see it in experienced nurses, right? They can be talking to you about 500 things and they're doing some complicated task and they're just like, okay, what, right? So that's a procedural memory. It's stored in your cerebellum. So when I was talking to you about how to learn muscle actions, for example, I said get it into your motor memory, put it in another part of your brain, right? Have some of it stored in your frontal lobe, have some of it stored in your parietal lobe and your auditory system and your visual system. Get it in as many parts of your brain as you can, because then if one of these circuits fails, you've got 5 others. Makes sense, right? And one of the reasons we know there's a difference between procedural memory and episodic memory is that patient HM that we talked about last week, the guy who completely lost his episodic memory, he couldn't remember how he started a sentence, right? He was still able to be trained in physical tasks like this one. So if the task was you're actually looking into a mirror, but you're drawing into the corners of a star, so it's a reversed image that you're actually looking at, you can't see your own hand directly. And the first time you try this task, everything's reversed and everything looks squiggly while you try to keep the line inside of those double lines, right? He did improve every day with that physical mechanical task. So that was part of how we determined there's a different area for your procedural memory, which I just think is cool stuff. I do have an article attached to this in your notes if you're interested in procedural memory and the difference between that and episodic memory. So get things into your motor memory. Advice for the next 3 years. All right, your textbook has a pretty goofy definition for consciousness, but we'll roll with it. We're not talking about philosophical consciousness, I think therefore I am. We're talking about being awake and aware of your surrounding. That's their definition for consciousness, which works just fine for our clinical purposes. We'll talk about level of consciousness in patho and how we're going to measure level of consciousness in relationship to cerebral pressure. Yeah, ICP, intracranial pressure, that's the word I'm looking for. So we'll define consciousness by being awake, experiencing the world, move about in it. Are your autonomic reflexes working? Do you have awareness? Do you have voluntary motor activity? And we're going to characterize it by the activity of the reticular activating system, which for our purposes is going to be the same as the reticular formation. This is a slide actually from my sleep lecture on the medical campus. And I actually like this image a lot better than your textbook's image of the reticular formation. It doesn't really help you at all. What the reticular activating system is, how we discovered it was we stuck a bunch of electrodes into a lot of rat brain stems and we measured which areas kept them awake. And it turns out there's a lot of different nuclei in your brainstem that help keep you awake. What this is showing you is that there's also a variety of neurotransmitters. This slide, again, is above and beyond what you necessarily need, but if you're interested in this in finer detail, you can see that acetylcholine, serotonin, norepinephrine, dopamine, histamine, those are all playing a role in keeping you awake and conscious. And it's by inhibiting these with GABA that we are allowed to fall asleep. So what you actually need to know for the reticular activating system is that the reticular formation is a scattered group of nuclei in your brainstem that project to your forebrain to increase your level of consciousness. So again, this is really important in terms of level of consciousness and why we have it. Which part? The reticular formation is a diffuse set of nuclei in the brainstem. It projects to the forebrain, releasing a variety of neurotransmitters to increase your level of consciousness. It also means your brainstem's really key to your level of consciousness, right? When we're monitoring your health after a brain injury, your brainstem's probably the most important part of that. Your level of consciousness is going to be a major indicator of your clinical outcome. Okay, and that's about it. Any questions? Yep.
So, you know when you take Benadryl, it's an antihistamine. Is that why it makes you sleepy?
That is the opposite of a dumb question. That is a perfect question. Yes, if we take an antihistamine, it's going to block this wakefulness pathway, and that's why it makes you drowsy. For some, I imagine so. It doesn't have that effect on me. Yeah, you can see that serotonin is part of a wakefulness pathway. So honestly, it's also part of a sleep pathway. Just for funsies, we're going to overcomplicate that. So, some people on serotonergic drugs are going to have wakefulness, and some people on serotonergic drugs are going to have drowsiness. Hooray! And that's about it. Any other questions before I close out this recording?